Assalamu alaikum wa rahmatullahi wa barakatuhu. Our episode today is to discuss blood-brain barrier. Hope you capitalize on it. Firstly, what's a blood-brain barrier? It's a highly selective permeability barrier that separates between the microvessel that surrounding the brain tissue and the extracellular fluid of the brain. Okay. What's the function of blood-brain barrier? Is prevention of toxins, pathogens, and potentially dangerous substances from crossing into the blood-brain barrier or circulatory system of the brain. Regarding the structural building of the brain, in the anterior part, it's composed of endothelial cells or microvessel, special type of microvessel, because their endothelial cells are connected by tight junctions. Tight junction between the endothelial cell of the blood vessel or microvessel of the brain, making them special vessels. Okay, and attached to the microvessel, the end foot process of pericyte or astrocyte. Okay, so we have the microvessel and we have the astrocyte, that is the most abundant CNS neuroglial cell. Okay. Another picture more closely illustrating the structure, the endothelial cells and the tight junction connecting them, and the end foot process of astrocyte. This is the astrocyte more closely illustrated. Astrocyte has photoprocesses. These are the projections. These projections attached to the microvessel and thus forming the blood-brain barrier. This slide showing the difference between the general capillary in our body and the capillary in the brain. The capillary in the brain is a special capillary, as we said, by the presence of the tight junction, and we have in general capillaries in our body we have halos or gaps that are called fenestration, that's separating the endothelial cells, okay, to allow passage of material and exiting of the material from the circulatory system. While here we have one layer, there is tight junction between these endothelial cells, okay, means that not any substance can cross into the blood system of the brain unless it matches the criteria. Specific criteria for the material are needed to in order to pass. Which substance to pass or not to pass? Okay, which substance allowed to pass inside the brain? Okay, cross the blood-brain barrier. Which substance is not allowed or it's halted from passing into the brain barrier, and the brain barrier will say stop, sorry, you are not allowed to go in. The substances that can pass or cross the blood-brain barrier, it is the nutrient, especially the glucose, that is the main source of nutrition to the brain, okay, to the brain cell. And we have the oxygen and we have the CO2, that is the water product, okay, can exit the brain tissue. Okay, the another criteria, it should be not too highly charged, not too large particle, and it should be lipid soluble. Again, it's not too highly charged, not too large, and it should be lipid soluble. That means it will go to the brain. So not if it is not matching these criteria, we will say stop. Okay, we have many factors that should open up the blood-brain barrier. They are microwaves, radiation, trauma, hypertension, and infection. Again, we have many factors that should open up the blood-brain barrier. They are microwaves, radiation, trauma, hypertension, and infection. We have certain structures in the brain that block the blood-brain barrier. They are the pineal body and the posterior pituitary gland, and we have the subfornical organ, and we have organum vasculosum of the lamina terminalis, and we have the subcommissural organ. Okay, why do these structures block the blood-brain barrier? Because especially for the pineal body and for posterior pituitary gland, pineal body, posterior pituitary gland, these are endocrinal gland. So endocrine gland means that they need access to the bloodstream or to the circulatory system, direct access to the bloodstream in order to secrete their hormones directly into the circulatory system. Okay, and lastly, thank you for listening.